All right, let's talk about section 15.3 now, applications of genetic engineering. So how have we made use of all of this technology that's been developed? Largely in the field of agriculture, this has happened, in particular with crops, with plants. This graph from the book shows over time the increase in certain crops and that have been genetically modified. And so this shows the percent of acres. So we can see the data is a little old, but it's probably increased even more. I would imagine you can see upwards of over 90% of soybeans, for example, that are planted in the United States have been genetically modified. And cotton's not too far behind, and the others you can see where they are. Um, these particular um, forms of these plants is the HT stands for herbicide tolerant. In particular, these plants have a gene introduced to them that came from a different species that makes them resistant to the herbicide called Roundup. So the farmers can spray the field to kill the weeds, but it doesn't kill their soybean or cotton plants. Um, the BT, these are plants that have a gene in them that came from a bacterium. So this is moving genes between different kingdoms. This gene gives them resistance to insects that try to eat them. Basically, the plants produce a compound, an internal insecticide, if you will, that when the insects eat them, it kills them. Um, if you can see corn is pretty high and has gotten even higher. If you eat, I don't know, Doritos or other kind of corn chips, there's a pretty good chance the corn that is in them has probably been genetically modified. Um, Really, the only way to avoid or being sure that your foods you don't eat, plant-based foods in particular, is to eat organic foods. Animals have not been modified as much. As I mentioned in previous videos, it's more difficult to carry out the procedure in animals. And some of the examples of this pig, this is sort of just a what you would call a proof of concept. It doesn't really give the pig any advantages or benefits really. They have a gene from a jellyfish in them that makes them have this sort of greenish color. Um, and with other animals like goats and cows, they're working on introducing genes in them such that in their milk they produce certain beneficial compounds that one could get by drinking the milk. Again, sort of more of an experimental phase there. All this modifying of these organisms is not without its controversy. There's some that think we shouldn't be doing this. It's sort of playing God with these things and going way beyond just traditional kind of breeding. But of course, the agricultural sector, they say it brings certain benefits in terms of productivity and certain beneficial traits in these plants and animals that could be useful for feeding a growing population. Here's another example with a plant. Rice, of course, is normally white and it has uh, a good number of calories in it, so it provides good caloric content, but it's not terribly nutritious. And so um, there are certain places in the world, certain countries where people eat a lot of rice, and so excuse me. So they may get all the calories they need, but they may be missing out on certain nutrients. And excuse me. So here is a genetically modified rice plant called golden rice that has a gene inserted in it from a different plant that allows it to produce vitamin A, which rice normally doesn't really produce in any amount at all, and that's what gives it its color. Vitamin A is necessary nutrient for giving us vision, among other things, and so this can be a problem in some parts of the world, and so this plant has been modified. You won't see it in the States because vitamin A deficiency is generally not a problem here. Um, Again, animals limited, but here's a link to a lab at the University of Iowa that works with genetically modified mice. And as far as I could tell from the website, they will produce genetically modified mice for you if possible. Um, Bacteria have been modified, as I think I mentioned earlier, in particular with these characteristics, these genes from people, that you have bacteria that will produce HGH, or human growth hormone, that the hormone can be extracted and then given to people. Same with insulin and blood clotting factors. These are all compounds that would normally be difficult to come about, but with these populations of transgenic bacteria that produce them, the compounds can be readily available and relatively inexpensive. Gene therapy, this is an experimental procedure to not change the genome, the complete genome of a person, but to try to change genes in certain cells that are not doing their job correctly. So because of some mutation, for example, with cystic fibrosis, your lungs produce an abundance of mucus, which causes problems. You can use a virus that the person might inhale, for example, and this virus has been engineered to have a good version of the gene, and viruses operate by injecting their DNA into a host cell. So the goal is to have the virus inject the good gene into someone's lung cells, and hopefully they will start making use of that gene in those cells. Um, this is still very much an experimental procedure and not really approved for general use. Of course, with genetic testing, we use these technologies as well um, with sort of what's called prenatal diagnosis, or basically before a baby is born. You can determine whether or not that baby in utero has certain detrimental characteristics, traits, mutations, whatever. If it's a trait that runs in the family, the couple that is expecting a child can go in and be tested um, to see if their baby has or is just a carrier of that disease. This is something I did right out of college when I worked in a lab. Some of these tests. Microarrays are is this technique to determine which genes are being expressed by a particular cell. Um, what you do is basically isolate DNA that represents particular genes. You attach the DNA for these different genes on this microarray, and each dot represents um, a particular gene. Um, and so you have this whole array of genes. This can have hundreds of thousands of genes on it, depending on how big the array is. And what you do is then, from the cell of interest, you'll isolate the mRNAs from that cell. And of course, the mRNAs are the products of genes that are being expressed by that cell. You basically see where the mRNAs attach, because the mRNAs will attach to DNA that they are complementary to. And the mRNAs have been labeled to fluoresce, or basically express a color. And so where you see the dot is lit up, that means that gene is being expressed in that particular cell. And for example, in the upper right corner, you can see there's a bunch of the dots that aren't lit up at all. They're just dark. So those would represent genes that aren't being expressed by this cell. In a different cell, if you tested the same genes, you might see that those are lit up because those genes would be expressed, whereas perhaps this one right here would not be expressed because it's not being used by that cell. In the example they give us, they're comparing um, two different cells, cancer cells and normal cells, on the same microarray, and they've labeled each with a different color. And so where the dot is kind of reddish or orange, that means it's a gene that's primarily being expressed in the cancer cell, where it's greenish, that means it's being expressed in the normal cell. If it's kind of in between, sort of a pale yellowish orange color, um, like perhaps this one right here, that's one that is being expressed by both types of cells. Sometimes you can just test one type of cell, or in this example, you can do a couple different kinds of cells if you like to. All right, I think we're going to stop right there. We're getting to DNA fingerprinting and forensics on the next video.